All right, let's do this. Broadcasting live all the way from Casa de Paulson. Let's talk about codominance. So we were talking incomplete dominance before. The big difference is you have two alleles, but they're going to be separately expressed. Okay, so this is different from the blending effect that we saw where you had kind of a medium in between the two alleles. Here you're going to see each allele has its own effect, and you'll see both of them at the same time. So the classic example of this is blood types. So for blood types, let me give you just a little background on blood typing. So blood cells, here's a blood cell, whoop, and they've got molecules on their surface. Your immune system works by recognizing the shapes of things that are on the surface of molecules. So there are actually three alleles in the case of blood types, but let's just focus on the first two. One is the A allele, and one is the B allele. And the difference between these two is that they code for what's called a glycoprotein. So protein, glyco means sugar. So this is a protein with some sugar attached to it. It's going to be placed on the surface, all around the surface of the red blood cell. And the difference is in the shape of the particular protein. So it's going to look different to your immune system. So if you have the IA type allele, that's going to put a particular shape. And so I'll just draw these A's here on the surface of this blood cell. Okay. Similarly, if you've got the B-type blood cell, you've got the B shape on your blood cells. Now, your immune system works by recognizing the shapes that are on the surface of foreign objects. So your body will produce what are called antibodies. These are little proteins who have a complementary shape to whatever's on the surface of foreign objects. So that means that if you are, let me just erase this, if you are if you are type A blood, here's A again, and I'm just going to draw up as triangles here. You've got these A-shaped glycoproteins on the surface, but it also means that you're going to have floating around in your bloodstream antibodies that will bind to the B shape. So here's a guy over here with the B shape. I'll draw them just as squares. Okay, so you have something that will fit and attach and actually stick to the other type of molecule that might be on the surface. Similarly, over here, our B is going to have something that would bind to the A shape. And if it sticks to it, it's going to cause it to clump and cause all sorts of problems as a defense mechanism. Okay? So that'll come into play when we look at the blood donation status. Okay, but let's get back to the genetics here. So let me clear this off. Okay, so again, we've got actually three alleles, the A, B, and both of these, they're codominant over this recessive one, the I type, which codes for nothing to be put on the surface. So the codominance comes into play if you have the genotype IA, IB. So your phenotype is you're going to have a blood cell with A's on it and B's. Okay, so it's not some shape that's a combination of an A and a B. You have A's separately all over the surface and B's separately all over the surface. So that's the codominance part. If you're, of course, if you're A, or AI, this is the dominance part, then your blood type is A. And you've got only the A shape, what we call these guys antigens. Only the A shape antigens on the surface. If you're B, or IB, your blood type B. And lastly, if you are I, that's the homozygous recessive type, you are type O. And conveniently enough, type O, this looks like a little naked blood cell with nothing on the surface, which is what it is. Okay, so let's do a problem here working with these genotypes. Okay, let's do that. Okay, so here we go. We got type A blood man marries a woman with AB blood. Okay, they're going to have some children, and we want to know what are the possible children they can have. So we work this just like any other problem. Type A blood, man. So let's make the Punnett Square here. We'll put the man up here. So type A blood. The one thing we don't know here is this guy heterozygous or homozygous? So we know that he has this allele because that's his phenotype. So this is his phenotype, type A blood. We actually don't know what's here yet, but we'll get to that. The woman, we know she's got an A here, and she's got to have the B allele also because she's AB. So we've taken care of her genotype. So let's look at what we can get here. A, B. We know that we're going to get a B allele and an A allele, but we don't quite know what's going to go here yet. This could be another A allele, or it could be I. Okay. So if it's an A, this person's going to be type A either way. They would be IA or IAI, but notice that this genotype, either way, leads to the same phenotype, A-type blood. Here's where it matters. We could either be AB again, or type B. Okay, so it depends on if this person is homozygous or heterozygous. So maybe that's not the best question, but anyway, gets you thinking about the possibilities. So let's real quick look then back to, get rid of that for you. Oh, there we go, make that disappear. Look at these different genotypes. Let's quickly talk about the donation status of blood types. Okay, so let's look at, here's our possible phenotypes. Let's fill in these genotypes right now. So we've got the genotype A blood. Okay, so you can do this along with me. Genotype A blood is going to be either IA, or IAI. Okay. Type B blood going to be similar to the B, or IBI. Here we got only IA, IB, here I and I. Okay, and so the antigen, this is the molecule that's on the surface. So if you are type A blood, then you have the A type molecules on the surface. If you're type B, you've got the B type. If you are AB, then you have both A and B. That's the codominance part, right? Separately expressed. If you're type O, you have nothing on the surface. You got a nude little blood cell. Okay. As far as the antibodies in the blood, these are the molecules that are floating around that bind to foreign objects. So if you're type A, it means you've got the B antibodies floating around in your body ready to bind to this molecule if they see it in your blood, because if it shows up, they know that it's foreign. Okay. Similarly, if you're B, then you've got the A-type antibodies. Now, if you're AB blood, you have both these shapes normally floating around in your blood on the surface of your blood cells. So you don't make any antibodies. On the other hand, if you're type O, you don't have either of those molecules, so A and B looks foreign to you, because you have the A and B antibodies floating around. So now this has some implications for donation. This molecule, the red blood cell from the type O with nothing on the surface, we can put that in anyone's blood because there's nothing for any antibodies to attach to. So this is the what we would call the universal donor. Because type O blood can go in anybody and no antibodies can stick to it. On the other hand, type AB blood, okay, because they don't have any antibodies, they can receive anyone's blood. It doesn't matter. They don't have anything to recognize A's or B's or nothing. So they are the universal recipient. Okay? They can take it from anybody, but if they give their blood to type B or type A, we're going to have a immune reaction, clotting, etc. Not good. So if you are type O blood, go donate some blood. Everybody can use yours. All right. That's it.